Метод превращения чего-то маленькими организмами, находящимися вокруг него, во что-то абсолютно другое, был известен человеку на протяжении столетий.
In biotechnology, essentially we are using the life processes of living organisms to produce a product.
You're just harnessing nature's ideas, nature's way of tackling various problems, and then using technology to bring that to the fore.
Biotechnology has endless applications.
This is the science of this century.
Микроорганизмы присутствуют везде. Эти простые клеточные организмы, которым более чем 3,5 биллиона лет, признаются самой старой жизненной формой планеты. Самыми многочисленными являются бактерии. В горсточке Земли можно обнаружить более 10 миллионов таких организмов. Бактерии известны также тем, что они приспособлены к жизни в любой среде и везде. Эти микробы, увеличенные в миллионы раз под электронным микроскопом, используются для того, чтобы разрушать самые токсичные вещества, известные человеку.
During first Gulf War and during recent Gulf War, there is a huge oil spills in the desert. When the oil spill will occur, it will spread. So whatever living thing is there in the water or soil, they will not get oxygen. So they will immediately die.
Группа ученых из Института по исследованиям в области энергетики, проведя многократные исследования и потратив 6 лет, разработала комбинацию из разных бактерий, которая может разрушить углеводород сырой нефти на более простое, а соответственно менее токсичное вещество – двуокись углерода. Этот коктейль они назвали нефтяной разрушитель.
Wherever oil contaminated site is there, from there we got these bugs. So we selected 4 different bugs and we make a cocktail out of these 4 bugs. Initially the response was not very encouraging. Then with the great difficulties, this Mathura refinery, they gave us the small area, and that was really mess. It was full of oil. Oil was floating on the land, and we applied these bugs, and within 1 month it was completely clean. Now in India we are supplying to all the oil industry.
Группа ученых из Терри нашла решение еще одной проблемы нефтеперерабатывающей индустрии. Обычно из нефтяной скважины извлекается только 40% нефти. Практически 60% нефти остается внутри каменных пород. Для удовлетворения нужд необходимо 82 миллиона баррелей нефти. Ученые института открыли также комбинацию микробов, способствующих добыче большего объема нефти.
So we have to inject these bugs with the high pressure, so these bugs should penetrate in the pores, and then in the pores they will produce the metabolic products, and they will also produce the gas. So they will create the pressure in the pores, and then they will displace the oil. So they will remove the oil, and then oil production will increase. There are such technologies available, but most of these technologies, they are working up to 60-degree temperature. But in the oil reservoir, 60-degree temperature, you will find only few reservoirs where temperature is 60. Whereas in our technology, we have developed up to 90-degree. And now we are upgrading our technology up to 120-degree, which nobody has so far.
В то время, как некоторые бактерии могут быть полезны для человечества, другие являются смертельными. Сибирская язва – заболевание крупного рогатого скота, но человек может заразиться ею при контакте с больным животным. И если не начать лечение своевременно, оно фатально. Бактерия, являющаяся причиной заболевания, называется бацилла антрасис, но наряду с токсином, вызывающим болезнь, она вырабатывает другую молекулу под названием защищающий антиген, которая предоставляет защиту от заболевания. Доктор Ракеш Бхатнагар с группой специалистов работает над вакциной против сибирской язвы. Они поместили гены, содержащие информацию о защищающем антигене, в безвредный микроб Е. коли, который обычно живет в пищеварительном канале человека. При точном разрезе генома вакцина не вырабатывает никаких токсинов, как при традиционной вакцинации. Это снижает побочные явления. Но самое главное, они разработали уникальный эффективный метод продуктивности.
We take this genetically engineered bacteria and put it in a bioreactor where we have optimized the conditions for its maximum production. And now this bug reads this information thinking that it belongs to the bug, and it starts making millions of molecules, which we call it protective antigen. It makes in 5-liter fermenter or bioreactor something like 10 million doses of this vaccine. The kind of productivity we have shown, that is 10 million doses in a 5-liter fermenter, was unprecedented. People are trying to put the gene into edible variety of plants. We are the first to express protective antigen gene of anthrax in plants. Since we are able to produce it 10 million doses at the laboratory scale fermenter of 5 liter, if the same technology is expanded to 500 liter fermenter, it can make something like 1 billion doses per day. And if the fermenter is run for 6 days, it can make 6 billion doses, which will be enough to immunize the whole world against this disease.
We are using different bugs for different products. It depends on the kind of vaccine we are developing.
We constantly had to look for new microbial bugs, so to speak, to look for new enzymes.
This is the richest place you can find all sorts of bugs. I have bugs which can do wonders in this country.
If you get more and more new bugs, you'll be able to develop more and more new molecules. The advantage for us today is all this can be patented.
So we stand a good 45 to 50% of the procurement for the UNICEF and other global institutions. So there's a phenomenal strength in the capacities that we have developed in India.
If it is science, we are among the top few. When it comes to patents, we are not pretty much there, but we are growing. When it comes to our ability, for example, to manufacture, we are second to none. And particularly, low-cost manufacturing is our forte.
We do have a very strong presence of ancillary industries which are supporting this industry, and that is why India actually has a global competitive edge in biotechnology in the manufacturing opportunities. For instance, we have very good engineering companies in India who are all developing state-of-the-art bioreactors.
Among the developing countries, I think we have the best scientific manpower and we have the best medical infrastructure in this country, out of the developing countries.
We tell every child to go for education. So that's good. That means we are a knowledge-driven society.
We have truly a global competitiveness when it comes to cost of innovation. I certainly believe that we have a very innovative scientific skill base. I think the world clearly recognizes that today's diseases are global diseases, so it's no longer confined to the developing world. There has to be partnering between the developing and the developed worlds.
We have two good, wonderful partnership examples. One is White, US, one of the big company in the vaccine field. They shifted the part of the manufacturing to our plant. This is the first time a multinational shifted a manufacturing base from a developed nation to a developing country.
Продолжается поиск возможностей снижения цен на производство вакцин. В Индийском научно-исследовательском институте ученые разрабатывают ДНК-вакцину. Гены, которые сражаются с болезнью, будут напрямую имплантироваться в человеческую дезоксирибонуклеиновую кислоту или ДНК, основной генетический жизненный материал. Это значит, что вместо того, чтобы вводить белки вирусов для борьбы с болезнью, человеческое тело будет само вырабатывать молекулы белков, чтобы преодолеть определенную болезнь. Эта технология значительно повлияет на цены вакцин.
One of the things that in this lab is developed is a DNA vaccine for rabies. A DNA vaccine can be kept even at 40 degrees, nothing will happen. And therefore you don't have to refrigerate. Not only for storage, even for transport purposes, you don't need refrigeration. Therefore the cost comes down significantly. There have been several adjunct technologies, but the main thing is actually the ability to transfer genes across species and sex. Really revolutionized this field actually. There's a tremendous amount of gene transfer activity is going on in nature, you don't see it. Therefore, the artificial genetic engineering process is not really new, except you are probably harnessing this gene transfer to a particular direction.
Возможность переносить гены вдохновила ученых всего мира увеличить подкормку урожая. Доктор Асис Дутта выделил гены амаранта, семена которого широко употребляются в пище. Эти гены назвали AMA1 и поместили в картофель. Это попытка сделать еду более питательной.
Now we got that GM potato. You can find that is a protein content is a 35 to 45% more protein compared to control. And also quality is very important, not only content of the protein. It's a quality that this protein has all essential amino acids quite high, particularly lysine and sulfur-containing amino acids, which are lacking in fact other cereals. GM potato one is much more nutritious.
If you have appropriate genes where plants can grow even when there is an adverse situation of a rainfall, that would make a tremendous impact to yield as well as to the economy of this country. We are not going to get more additional land for cultivation. More or less we have reached cultivable land has reached a saturation level. And water, the more increase in land, you are going to have more requirement for water. That's going to be the biggest limitation in future.
Генетический материал для всех форм жизни одинаков. Хромосомы присутствуют во всех клетках и состоят из ДНК. ДНК, в свою очередь, можно расчленить на части, известные как A, T, G и C. Эти 4 простые составные могут соединяться, образуя пары. Знание точной последовательности этих пар оснований – необходимое условие, которое помогает ученым идентифицировать гены. На международном консорциуме 10 стран определили последовательность геномной структуры риса. Рис состоит из 12 пар хромосом, и задача индийских ученых – описать точную последовательность комплементарных пар 11-й хромосомы.
The total number of the bases in rice are 430 million bases. You can see that if we start to write those bases or letters, how thick would be a book. What I know is that in this book, India has written a chapter in which there are about 12 million letters. So that is Indian contribution. By sequencing, we know the genes. So we can select which genes are responsible for a particular trait. And then we could look for better genes. Recently, after screening rice genes, we got a gene which is induced by several stresses: salt, cold, drought, like that. So we took that a gene and placed in a model plant, that is tobacco, and tried to see can it make tobacco plants which are engineered now tolerant to these stresses. And we found that yes, that is possible. Now what is important is all the experiments which we have performed are still in our laboratory or in greenhouse. We have to still see that how they will perform in the field. India is a country which has a maximum area under cultivation of rice. We grow it almost on 40 million hectares. We have a great germplasm available, large number of variability in rice, including some of the wild rice that we call, which has got very useful genes, which can be engineered and which can be investigated by this. Even if we make 1% improvement in the yield, it will be a tremendous achievement.
Еще один золотой слиток, который является центром активных исследований индийских биотехнологов, это бомбикс мори, более известный как тутовый шелкопряд. От яйца до яйца цикл жизни шелкопряда составляет 60 дней, на протяжении которых он увеличивается в размере и в весе в 10 тысяч раз. В определенный момент он, для того чтобы защитить себя во время трансформации, вырабатывает драгоценный для человека продукт – шелк. Понимание генетикой того, что производит шелкопряд, помогло ученым выбрать те свойства, которые улучшают качество и производство шелка. Мотыльки с разными характеристиками тщательно отбираются и спариваются.
Conventionally, silkworm are being improved for many years, hundreds of years now. But biotechnology is to do precision breeding, to add precision to the existing conventional technologies.
We have integrated both classical breeding and molecular biology. As a result of this, we have already developed 3 hybrids. One is Swarnandhra, another is Kalpatarvu, and the third one is Hemavati. So the product developed by adopting this DNA technology and classical breeding, the cocoon characters are superior when compared to the traditional hybrid, and the silk yield what we are getting, the silk quality what we are getting from this hybrid is international grade silk.
Шелкопряд имеет также другую функцию. Это биореактор мини-предприятия по производству протеинов. Так как его тело превращает протеины шелковицы в шелковый протеин, биотехнологи используют эту характеристику для других протеинов.
Because silkworm is a domesticated insect and it can be easily grown and it increases in size by 10,000 times. So if you introduce anything inside the silkworm, it can produce, instead of the silk protein, it can produce this protein by so many times.
Знание генетической структуры какого-то вида помогает ученым понять основные жизненные функции. И целый современный мир стремится к тому, чтобы понять устройство звеньев целой цепи, от ДНК до протеинов. Полученная информация вызывает острый интерес.
If you stretch out DNA in each cell, it would be approximately about 2 meters. So if you take all the DNA which is contained in human body and join end to end as a stretched polymer, it might cover distance from the Earth to the Sun.
У человека 23 пары хромосом, состоящие из более 3 биллионов пар оснований. Сегодня ученые знают точную последовательность и расположение 3 биллионов пар оснований человеческого генома. Эта информация предоставляет невообразимую возможность познать причины и взаимосвязи нарушений и болезней. Арджуну 8 лет. Он страдает, вероятно, самым распространенным в мире генетическим нарушением крови – талиссемией. Обычные красные кровяные тельца получают свой цвет от разных пигментов или гемоглобина, который они переносят, который, в свою очередь, переносит кислород к разным частям нашего тела. Кровь Арджуна выглядит так же. Его костный мозг вырабатывает красные клетки, но из-за генетического нарушения в клетках мало гемоглобина, и в действительности они пустые.
The gene for producing hemoglobin is present on chromosome 6 and chromosome 11. One is the alpha globin and the other is the beta globin. In the beta globin gene, just one letter or one base changes from G to C, and that causes such a devastating disorder, which means that the genetic material has to be so precise if it is to function normally in an individual. The advent of molecular testing, DNA testing, has made all this possible by offering us the possibility of a very precise diagnosis. We can use that diagnostic precision to pick up the disease in a cell even without the individual actually being born. So antenatal diagnosis is the next product. The cost of doing diagnostics for just confirming hemophilia would be 1/20th in India as opposed to the United States.
Еще одной областью исследований индийских ученых являются стволовые клетки.
Stem cells are such cells which have two properties. They self-regenerate. You can make more stem cells out of stem cells, hence the word stem. And the second, you can take them and direct them largely to whatever tissue you might actually want into other cell lines. We take a little bit of the stem cells from one portion of the eye, grow it on an amniotic membrane to make a tissue which is stitchable, transparent, and will restore vision to the patient. This is called the limbo stem cell culture. In fact, we happen to be the world's largest human trial, successful human trial of adult stem cell technology. Over 200 patients, 70% success.
Но самое большое достижение медицинских исследований – понимание человеческого мозга.
We earlier thought of the brain as a static organ that decayed with age. Today we know that it's a dynamic organ that transforms itself to meet the needs. So as we use a certain part of the brain more, the connections are strengthened in that part of the brain, and what we don't use is pruned away. The more we use, the better it gets. Anything is possible with the brain.
Имея массу, равную 1,5 килограмма, человеческий мозг содержит 100 биллионов нервных клеток, образующих сложные для понимания миллионы биллионов связей. Если нейтрон мозга разрушен в результате какого-то нарушения, наблюдается приобретенное слабоумие. Доктор Панкадж Сетх разрабатывает причины гибели нервных клеток, вызванных самым смертельным вирусом в мире – вирусом СПИДа. Доктор Сетх разрабатывает версии, как штамм С вируса СПИД движется по частям тела, разрушает гематоэнцефалогические барьеры и использует мозг как резервуар. При этом заражаются разные клетки и разрушается нейтрон. В то время как ученые всего мира много знают о штаммах A и B, штамм C остается недостаточно изученным.
90% of the AIDS population in India have the strain C. That's how we are different from the developing countries or from countries like USA and Europe, where they have the strain B. As of now, 50% of the world population is affected by strain C. At National Brain Research Center, we are working on strain C virus, trying to understand how this virus causes the pathogenesis in the brain and how is it different from B.
Сегодня для того, чтобы понять совокупность биологической системы, ученые обращаются ко многим отраслям знаний, от математики до компьютерной науки, а также руководствуются традиционной медициной.
Parkinson's disease was described in ancient Ayurveda, and a plant that was used for that, for Parkinson's disease treatment, today we know it contains the same active drug that is given to Parkinson's disease patients today, which is L-DOPA. The interesting thing is, when you give the plant material as a whole to the Parkinson's disease patients, they don't have the side effect that you would see with the individual compound that's given in modern medicine.
There is a wealth of traditional knowledge here, and because drug discovery has become so expensive now with a failure rate of 99%, going back to your traditions and discovering new uses of old and traditional medicines is a very powerful opportunity.
Принимая во внимание традиционную медицину, ученые из Индийского института химической биологии и в Devs Medicals используют современные методы для того, чтобы проанализировать свойства определенных трав. И они обещают найти решение для многих заболеваний, причиной которых является стресс.
If you ask which is the major medicine on the counter, sold all over the global scenario, it is antacid. The reason is antacid is sold because there is acidity in the stomach. And why acidity is there? Because of the stress. So if you can combat the stress, you can combat the acidity also.
Tresina is a herbal product. It's a polyherbal formulation based on Ayurvedic principles. And we believe it should be able to combat stress. It can be internal or external, which adversely affects the system. We have been trying to establish Tresina as an anti-stress agent in the light of today's knowledge.
Если ранее наблюдали только эффект какой-то продукции, биотехнология предоставила ученым возможность понять механизм его действия, почему и как он в действительности действует.
We are observing that what is the mechanism, how Tresina is acting better than the others. That's why we are separating different compounds, wanting to see what is in this Tresina, what is more making synergistically more active. Until and unless we do it, the global acceptance will be not better.
Herbal medicines can be an alternative to modern medicines, especially in cases of chronic disease segments. A person is diagnosed to be diabetic at the age of 30, and he has to live till 80. So for 50 years, he has to have a medication, allopathic medication. And there are adverse effects. They become refractory, like you need higher doses to get the same effects. And in those areas, herbal medicines could turn out to be very good alternatives, singly or in combination with allopathic drugs.
Задача ученого мира сегодня – найти новые и лучшие лекарственные препараты. Знание ген и их функций придает точность поискам. Ставится вопрос о том, что медикаменты должны быть предсказуемы и адаптироваться к каждому индивидуально. Но до сих пор так много остается невыясненным. Причина, почему один человек более восприимчив к заболеваниям, чем другой, может заключаться в 0,1 части процента одного генома, который состоит из 3,2 биллионов пар оснований. И это тяжелая задача для ученых всего мира – проанализировать данные и найти причину этой разницы.
When we study genetic diseases, when we sample families, we need to get blood samples from members of these families. I've worked in the US, and members of families are dispersed all over the United States oftentimes, and therefore to be able to collect blood samples from members of the same family becomes very difficult. One may need to travel several thousand miles. Here we have this advantage that members of a family stay together, and that's a major advantage.
India has a strategic advantage because to identify the variation, we need to have large population, large families, and large number of children per family. You can see the way our marriage pattern that takes place. Our marriage pattern decides an endogamy, and we have this endogamous group who are isolated populations. And thereby we have a unique opportunity in this area to look for gene discovery, which is a very hard job.
We are trying to leverage the cutting-edge technologies in computer science and software to build very specialized tools that can improve the efficiency and the success rate of finding new drugs.
Bioinformatics requires mastery of 3 components: knowledge of computers, knowledge of biology, and knowledge of mathematics and statistics. India probably has more people that fit this bill than anywhere else in the world. India is making its mark in bioinformatics, and the investment required for the field is very limited. You need computers, you need storage, you need networks. This infrastructure is already coming up because of the IT boom happening in the country. So I only see a future that is going to get brighter.
In the last 10 years, we have heavily invested in putting in our universities and research institutes some equipment. this century, but I think many next centuries will belong to biology and biotechnology.